In 2013, a genetic study found that one tiny change in the human EDR gene was associated with more active sweat glands in Han Chinese volunteers. The variant was already famous for affecting hair and teeth. Suddenly, it was pointing towards something far more ancient, the way human skin handles heat. That discovery seemed to offer an extraordinary possibility. Perhaps the surface of the body still carries a measurable record of climates survived by distant ancestors. But there was an immediate problem. The visible pores on your skin are not a simple map of sweat glands, and sweat gland density is not a metabolic fingerprint. Two people can appear to have very different skin texture while possessing overlapping thermoregulatory capacity beneath the surface. And even when gland densities differ, the number of glands alone does not tell us how much a person will sweat during heat or exercise. What matters is which glands become active, how strongly each gland secretes, where those glands sit, how large the body is, and how efficiently sweat evaporates. That turns a simple ancestry story into a deeper biological puzzle. Human skin does preserve evolutionary history, but not in the way a pore counting app could reveal. To understand the real signal, we have to separate three things that are often confused: pores, eccrine sweat glands, and metabolic rate. The distinction leads back millions of years, to a transformation that made the human body unusually dependent on bare skin and evaporative cooling. Most mammals do not cool themselves by flooding nearly their entire body surface with watery sweat. Humans are extraordinary in that respect. Across most of our skin are millions of eccrine glands, tiny coiled organs that can push water and electrolytes through ducts to the surface. PubMed Central, PMC. When that water evaporates, it carries heat away. The system is so effective that a human can sustain prolonged activity in conditions that would rapidly overheat many mammals. The mystery is not whether sweating mattered in human evolution. It did. The harder question is why modern people differ in gland density, sweat output, and heat response. Some of those differences are developmental, some are environmental, some reflect body size, some are genetic, and a few may preserve traces of population history. PubMed Central, PMC. That 2013 EDAR result was one of the clearest genetic clues. The variant, known as VIC370A, became common in East Asian and Native American ancestry and has strong evidence of past positive selection. In laboratory models, the derived version of EDAR alters several ectodermal traits at once. Hair becomes thicker. Tooth morphology changes. Mammary gland branching changes. Sweat gland number can rise. PubMed Central, PMC. In humans, researchers found that individuals carrying the derived allele had a higher density of active eccrine glands on the fingertips than people without it. This matters because EDAR is not a sweat gene in isolation. It participates in developmental signaling that helps build several structures derived from the embryonic ectoderm. One mutation can therefore leave a package of effects across hair, teeth, glands, and other tissues. Evolution can act on that package, even if only one component is strongly advantageous. PubMed Central, PMC. That is called pleiotropy, and it makes reconstructing the original selective pressure difficult. A variant may spread because of one trait while dragging several others with it. For EDAR V370A, scientists have proposed multiple explanations. Increased sweat gland density could have mattered, so could effects on mammary tissue, hair, or other linked traits. One hypothesis places part of the selection story in the cold, dry conditions of northern Asia during the last ice age, rather than in a simple hot climate scenario, PNAS. That sounds paradoxical. Why would a variant associated with more sweat glands become common in populations living through extreme cold? Because sweat glands are not the only phenotype controlled by the pathway, and because the same developmental change may influence physiology in more than one direction. The Ice Age hypothesis argues that altered mammary ductal branching could have influenced the transfer of nutrients to infants under conditions where vitamin D deficiency was a serious problem. PNAS. It remains a hypothesis, not a solved case. The important point is that modern gland density cannot be read backward as a precise thermometer of ancestral climate. Evolution rarely leaves such clean labels. Populations migrate, mix, encounter new environments, and carry variants whose original advantage may no longer be obvious. Even the term pore density hides several biological structures. A visible facial pore is usually associated with a hair follicle and sebaceous gland, especially in oily regions of the face. Eccrine sweat ducts also open onto the surface, but their openings are microscopic, and their distribution varies dramatically by body region. Palms and soles contain some of the highest eccrine gland densities in the body, yet these areas tell us little about the facial pores people notice in a mirror. The trunk and limbs contain lower densities, but their enormous surface area makes them central to whole body cooling. PubMed Central, PMC. Humans possess roughly a few million eccrine glands, and the total number is established early in life. As a child grows, the skin expands around that largely fixed inventory. This creates a simple but important consequence. Larger bodies tend to have fewer glands per unit area because the same basic gland population is spread across more surface. PubMed Central, PMC. A person can therefore show lower gland density without possessing a different ancestral thermoregulatory strategy. Growth alone can change the measurement. Sweating performance also depends on secretion per gland. A region with fewer active glands can still produce substantial sweat if each gland has a high output. Conversely, a densely packed region can contribute little to heat loss if the glands are weakly stimulated or if the secreted sweat cannot evaporate efficiently. This is why researchers measure more than gland counts. They examine sweat onset, local sweat rate, active gland density, output per gland, core temperature, skin temperature, and environmental conditions. Once those variables are measured together, a striking pattern appears. Heat adaptation is highly plastic. The body can change its sweating behavior within days. Repeated exposure to heat can cause sweating to begin earlier, increase the total sweating response during exercise, expand plasma volume, and improve cardiovascular stability. PubMed. Sweat can also become more dilute as the glands reabsorb more sodium, allowing the body to conserve salt during prolonged heat exposure. These changes occur without generating a new population of sweat glands. The hardware remains largely the same. The nervous, cardiovascular, and glandular control of that hardware changes. That fact alone undermines the idea that pore density reveals a fixed metabolic destiny. Two genetically similar people can develop different heat responses after living in different climates. PubMed Central, PMC. And two people from different ancestral backgrounds can converge physiologically when they train, work, or live under the same repeated thermal stress. Long-term tropical exposure adds another layer. Some studies comparing people raised in hot, humid regions with people from temperate climates have found slower or reduced sweating responses in tropical residents. At first, this appears backward. If sweating cools the body, why would generations or years in the tropics produce less vigorous sweating? PubMed. The answer lies in efficiency. In humid air, sweat that drips from the skin removes far less heat than sweat that evaporates. Producing enormous volumes of liquid can therefore waste water without delivering proportional cooling. A restrained response can sometimes be advantageous when evaporation is limited. Humans are not merely sweat-producing machines, they are water-budgeting organisms. Every milliliter secreted onto the skin came from body fluid that must eventually be replaced. PubMed Central, PMC. In environments where heat is intense but drinking water is uncertain, thermoregulation becomes a competition between immediate cooling and future dehydration. This is the point where the title's idea of metabolic efficiency becomes more interesting, but also more precise. Sweat gland biology can affect the cost of shedding metabolic heat. It does not directly reveal how many calories a person burns at rest. Basal metabolic rate depends heavily on body size, lean tissue, organ metabolism, age, hormones, and other factors. There is no validated rule allowing someone to count skin pores, infer ancestry, and then calculate an inherited calorie preservation setting. What skin can reveal is part of the thermal interface through which metabolism meets the environment. Every calorie transformed into work also produces heat that must eventually leave the body. During exercise, muscles are inefficient converters. Much of the energy they consume becomes heat rather than mechanical motion. As intensity rises, blood transports that internal heat toward the skin. Cutaneous vessels dilate. Sweat glands receive cholinergic signals from the sympathetic nervous system. The secreted fluid reaches the surface, and if the surrounding air can accept more water vapor, evaporation removes heat. The performance of that system depends on climate. Dry heat rewards evaporation. High humidity blocks it. Wind accelerates it. Clothing can trap it. PubMed Central, PMC. Body geometry also matters. A tall, slender body has more surface area relative to mass than a short, broad body of the same mass. Across human populations, body proportions show broad associations with ancestral climate that are consistent with well-known thermoregulatory principles. Longer limbs and relatively slender builds can increase surface area available for heat exchange, while more compact forms reduce surface area relative to volume and can conserve heat. PubMed Central, PMC. But these are population-level patterns with enormous overlap. They do not justify identifying an individual's ancestry from silhouette, skin texture, or sweating behavior. Modern humans are genetically mixed, environmentally mobile, and behaviorally flexible. Air conditioning, clothing, housing, hydration, occupation, and fitness can overpower many subtle inherited differences. The same caution applies when researchers compare ethnic groups in a heat chamber. Ethnicity is not a single biological variable. It can bundle ancestry, childhood climate, diet, socioeconomic conditions, acclimatization history, body composition, cultural practices, and current fitness into one label. If those factors are not controlled, a difference in sweating can easily be mistaken for a genetic adaptation. That is why some reviews of human heat adaptation argue that many reported group differences can be explained by differences in opportunity for acclimatization rather than natural selection. PubMed. Yet genetics cannot be dismissed either. EDR shows that variants affecting eccrine gland density exist and can differ strongly in frequency among populations. The challenge is determining what those differences mean functionally. More glands do not automatically equal better heat tolerance. Whole body sweat rate equals, in simplified form, the number of active glands multiplied by the average output of each gland across the sweating surface. PubMed Central, PMC. A population may have higher active gland density in one region, but lower gland output, delayed activation, or different distribution elsewhere. In a 2017 comparison of young men of African ancestry and Korean men living under controlled conditions, researchers measured sweat responses across 8 body regions during passive heating. The groups differed in the timing of sweat onset, active sweat gland density, and output per gland across regions, but the pattern was not a simple hierarchy of one group cooling better. PubMed. The African participants showed a smaller rise in mean body temperature during the passive heating protocol despite later sweat onset. That result demonstrates why a single variable can mislead. Thermoregulation is an integrated system involving skin blood flow, body morphology, heat storage, sweat distribution, and evaporation. A lower sweat rate can sometimes accompany effective temperature control if heat transfer is being accomplished through another combination of mechanisms. PubMed. A higher sweat rate can be inefficient when much of the fluid runs off instead of evaporating. The body also changes its strategy depending on whether heat originates outside or inside. Sitting in a hot room is not physiologically identical to running in that room. Exercise generates substantial internal metabolic heat. Passive heating transfers energy inward from the environment. Both raise thermal strain, but they recruit the system differently. Fitness modifies the response as well. Endurance-trained people often begin sweating at lower internal temperatures and can achieve higher sweat rates during exercise. That does not mean training creates more eccrine glands. It improves how existing glands are recruited and how cardiovascular systems support heat dissipation. Age works in the opposite direction. Older adults can show reduced sweating and skin blood flow responses, increasing vulnerability during extreme heat. PubMed Central, PMC. Again, the gland count does not tell the entire story. Neural control, gland responsiveness, cardiovascular capacity, medication use, and health conditions can all matter. Sex-related differences can appear in sweat output as well, especially when comparisons are made at fixed workloads or fixed absolute heat production. But body size and metabolic heat production can explain substantial portions of apparent group differences, which is why modern experiments often normalize carefully for these variables. PubMed Central, PMC. This brings us to caloric preservation. In cold climates, metabolic heat is valuable. In hot climates, it is a liability that must be dumped. Humans can adjust heat production through behavior, shivering, activity, clothing, and longer-term physiological adaptation. Resting metabolism can vary among individuals and environments, but tying those differences directly to skin pore density overstates the evidence. Sweat glands are an output system for heat. They are not the master regulator that sets basal energy expenditure. Another subtle variable is the maximum output of each gland. Glands are not identical pumps with fixed flow rates. Their secretory capacity differs among individuals and body regions, and repeated heat exposure can increase functional output. That means two people with similar anatomical gland density may display very different whole body sweat rates once core temperature rises. Anatomy sets part of the ceiling, but physiology decides how much of that ceiling is used. PubMed Central, PMC, the chemistry of sweat matters too. Eccrine glands first produce a fluid whose composition is closer to extracellular fluid, then reabsorb sodium and chloride as that fluid travels through the duct. When flow becomes very high, there's less opportunity for complete reabsorption, so sweat sodium concentration can rise. With heat acclimation, sodium conservation often improves. In resource-scarce environments, preserving salt while sustaining evaporation could be as important as producing large volumes of sweat. PubMed Central, PMC. Water availability creates an evolutionary constraint that laboratory heat chambers can easily hide. In an experiment, participants usually begin hydrated and can drink according to protocol. Ancestral environments were not controlled that way. A thermoregulatory strategy that performs beautifully for 1 hour with unlimited water may become dangerous across an entire day if streams are distant, food is scarce, or humidity prevents efficient evaporation. Metabolic heat also scales with activity and body mass. A larger person moving at the same relative intensity can generate more absolute heat than a smaller person. This is one reason comparisons based only on ambient temperature can mislead. The thermal problem is not simply how hot the air is, it is how much heat the body is producing, how much surface is available to exchange it, and how much evaporative capacity the environment allows. This helps explain why researchers increasingly interpret population differences through integrated heat balance rather than isolated traits. A gland density difference may be biologically real, while contributing only modestly to whole body outcome once morphology, fitness, acclimation, and environmental physics are included. The more variables are controlled, the harder it becomes to defend sweeping claims about one ancestry being inherently more thermally efficient than another. The evolutionary connection is therefore indirect. A lineage living for many generations in a harsh climate can experience selection on many traits simultaneously. Body proportions, pigmentation, hair characteristics, vascular responses, fat distribution, diet-related metabolism, immune function, and skin appendages can all face different pressures. Some adaptations are genetic, others are developmental, others are reversible acclimatizations, and some apparent adaptations may simply reflect population history or genetic drift. The skin is remarkable because all of these forces can leave marks in the same organ. Pigmentation responds to ultraviolet radiation. Hair follicles change insolation and solar exposure. Eccrine glands govern evaporative cooling. Blood vessels regulate dry heat transfer. Subcutaneous tissue influences insulation. Sensory nerves detect temperature before the core is in danger. Yet these traits do not evolve as one coordinated climate package. Different genes have different histories, and selection can act on one trait without optimizing every. HEDAR V370A is the perfect example. It is tempting to tell a clean story in which hotter ancestry produced more sweat glands, but the alleles, geography, timing, and pleiotropic effects make that explanation incomplete. Its rise may have involved cold climate pressures, infant nutrition, or a combination of selective advantages. The variant appears to have originated more than 20,000 years ago, and its frequency increased dramatically in ancestors of many East Asian and Native American populations. PNAS. Ancient DNA studies have helped trace this history, showing how a developmental allele could spread across populations long before modern national or ethnic categories existed. That time depth matters. The people carrying it today inhabit climates ranging from tropical forests to Arctic regions, megacities, deserts, mountains, and climate-controlled buildings. The allele has not disappeared simply because the environment changed. Evolution does not reset when a family moves, but neither does the allele dictate an individual's heat tolerance. One variant is only a small part of a system involving thousands of genes and a lifetime of exposure. This is why ancestry inference from skin function must remain probabilistic. A trait can be statistically associated with a population without being unique to that population. Migration and intermarriage move alleles across boundaries. Natural selection changes frequencies, not identities. Individuals inherit shuffled combinations, not ancestral climate labels. Even within one person, sweat gland density varies dramatically by site. The forehead, back, forearm, palm, thigh, and sole are not interchangeable measurement surfaces. Regional sweat output also changes with exercise, acclimation, posture, and local skin temperature. A measurement from 1 square centimeter cannot automatically represent the whole body. PubMed Central, PMC, this becomes crucial when consumer technology claims to read health or ancestry through skin texture. High-resolution cameras can estimate visible pore appearance, but that is not equivalent to histologically counting eccrine glands. Sebum production, age, sun exposure, acne history, follicle size, cosmetics, hydration of the outer skin, and imaging conditions can all alter how prominent facial pores appear. So the biological blueprint is real, but it lies deeper than the photograph. To map thermoregulatory ancestry, researchers need genetics, physiology, controlled heat exposure, and population history together. A serious study would measure genotype, active gland density across standardized regions, sweat rate per gland, body surface area, core temperature, skin blood flow, fitness, and acclimatization. It would also need participants whose childhood environments and recent thermal exposures are carefully documented, because the nervous system can remodel heat responses remarkably quickly. Then the experiment would have to separate ancestry from geography. Someone with East Asian ancestry raised in Northern Europe may not respond, like someone with similar ancestry raised in tropical Southeast Asia. A migrant study can be especially revealing because it separates inherited variation from years of environmental conditioning. When researchers have compared tropical natives, temperate residents, and migrants, they have repeatedly found evidence that acclimatization can change sweating patterns without changing ancestry. That plasticity may itself be one of the most important human adaptations. Instead of evolving a perfectly specialized body for every climate, humans evolved systems capable of adjustment. PubMed Central, PMC. Sweating is a powerful example. The number of glands is largely built during development, but the amount they secrete can change with training and heat exposure. The threshold for activation can shift. Sodium conservation can improve. Plasma volume can expand. Cardiovascular strain can fall at a given workload. PubMed Central, PMC. This allows one anatomical platform to operate under multiple climates. It is not infinite flexibility, but it reduces the need for a new genetic solution every time the environment changes. From an evolutionary perspective, that may help explain why modern humans could disperse across such a wide range of thermal environments while remaining one species. Culture multiplied the effect. Fire, shade, clothing, shelters, water storage, altered activity schedules, and eventually mechanical cooling changed the selective landscape around the body. A person no longer had to survive midday heat using physiology alone. Behavior could protect the same genome from conditions that would otherwise impose severe thermal stress. This creates another reason that modern metabolic differences should not be interpreted through ancestry too quickly. The environment acting on our bodies is increasingly self-constructed. Two neighbors with very different ancestral histories may spend their days at the same indoor temperature, eat similar diets, perform similar work, and exercise in the same gym. Their resting energy expenditure may differ, but those differences cannot be assigned to sweat gland density without direct evidence. The relationship between climate and metabolism becomes clearer at the whole body level. Heat balance depends on metabolic heat production minus external work, balanced against dry and evaporative heat exchange. If heat production exceeds heat loss, body temperature rises. If heat loss exceeds production, body temperature falls. Sweat glands sit on the loss side of that equation. They help dispose of heat already produced. They do not determine the entire production side. That distinction resolves the central contradiction. Skin can preserve clues to ancestral thermal biology without serving as a meter of ancestral calorie thrift. The efficiency visible in thermoregulation is often about conserving water and salt while removing enough heat, not about minimizing basal calorie expenditure. In dry conditions, high sweat capacity can support extraordinary evaporative cooling, provided water is available. In humid conditions, the optimal response may involve limiting wasteful sweat, increasing skin blood flow, reducing activity, seeking airflow, or shifting activity to cooler hours. Resource scarcity changes the equation again. Dehydration reduces plasma volume and threatens both circulation and sweating. A body that loses water too rapidly may cool effectively for a short time but become vulnerable later. A body that sweats too little may conserve water but overheat sooner. There is no universal best setting. The successful phenotype depends on air temperature, humidity, solar radiation, wind, clothing, activity, body size, and access to water. Evolution therefore operates on trade-offs, and trade-offs are exactly what a single pore density number cannot capture. The most ancient signal is broader than ancestry. Humans as a species possess an exceptional density of eccrine glands across hairy body skin compared with other primates and mammals. That expansion was part of a major thermoregulatory transition associated with reduced body hair and improved capacity for sustained activity and heat. PANAS. Developmental research has begun to identify regulatory changes that helped make this human pattern possible, including changes in pathways controlling sweat gland progenitors. In other words, before there were population differences in gland density, there was a much larger species-level transformation, PANAS. Every modern human inherits that foundational cooling architecture. The differences among populations sit on top of a shared human system that is already extreme by mammalian standards. This perspective shrinks some ancestry differences while making the evolutionary story more impressive. The greatest thermal divide is not between modern populations. It is between humans and the ancestral mammalian condition from which our sweating system evolved. Only after that transformation do variants such as EDAR V370A become meaningful as local modifications of a broadly shared platform. That is why the pores on your skin can be called historical only with care. Some of the structures beneath them were shaped by ancient selection. Their density can be influenced by inherited developmental pathways. Their performance can be altered by a lifetime of environment. Their visible appearance can be dominated by completely different features, such as follicles and sebaceous glands. And their relationship to metabolism is functional rather than diagnostic. Metabolism produces heat, while skin determines how effectively much of that heat can escape. The 2013 EDAR finding, therefore, did not reveal a hidden calorie code written across human skin. It revealed something more scientifically valuable. A single developmental variant could measurably alter part of the body's cooling hardware and preserve evidence of recent human evolutionary history. The studies that followed made the picture less simple, not less powerful. They showed that selection may have acted through several traits at once. PubMed Central, PMC. They showed that climate adaptation cannot be reduced to a hot versus cold slider. They showed that acclimatization can imitate or overwhelm inherited differences. And they showed that the skin is not a static fossil. It is an active organ whose inherited structure is continuously reinterpreted by nerves, blood flow, fitness, hydration, and environment. PubMed Central, PMC. So if two people stand side by side in the same heat, Their bodies may begin sweating at different moments and distribute that sweat differently. Those differences may contain traces of genotype and ancestry, but also traces of childhood climate, recent training, body size, age, and the previous weeks of weather. The skin remembers evolution, development, and experience at the same time. That is the real hidden blueprint beneath human pore density, not a map that tells us exactly where our ancestors lived or how efficiently they burned calories, but a layered thermoregulatory system built by selection and rewritten by life. Our pores do not reveal a metabolic destiny. They reveal something more human, the ancient capacity to survive by changing how we release the heat we create.